In this video tutorial, we will be discussing about the monocot root. So in the previous video, we completed the dicot. So today we'll go with the monocot. So all the monocot and dicot shows the similarities, but only at the vascular bundle. So we will be drawing those vascular bundles now. So as we discussed, in the dicot root, it will be showing strands of xylem. So here the protoxylem is towards the peripheral side, whereas the metaxylem is towards the center side. So in the dicot root, we have seen the 4 strands of arrangement of this xylem strands. But in the monocot, we have more than 6 to 8 strands of arrangement can be seen. There we call it as a tetrarch. Here we call it as the polyarch. But the facing is not changing. All the protoxylem is towards the peripheral side and the metaxylem is towards the central side. Only the difference is the number is increased. So here this is the protoxylem and this is metaxylem. Protoxylem is towards the peripheral, metaxylem is towards the center. So that's why it is the exarch type of xylem. As more than 6 are present, so we are calling this type of arrangement as polyarch type. All these are on the same strand, and when we are seeing the phloem is present alternate to the xylem. So let us draw these are the phloem. They are just not on the same radius, they are separated, they are not jointed, but they will be joined by a conjunctive tissue. So here we can find patches of or are not continuous in between the strands of xylem. These structures are the phloem. We know that the function of phloem is for the conduction of food. In between these two, in between the strands of this xylem and phloem, you can find a conjunctive tissue. So in the dicot, we have seen the conjunctive tissue is of the parenchyma. So initially, even in the monocot, the conjunctive tissue is parenchyma. But whenever the plant is growing, during the older stages, this parenchyma will be replaced with the sclerenchyma just to provide the extra support, because we know that monocot roots doesn't show any secondary growth as the dicots because there is no cambium formation. So this whole yellow colored cells which are connecting the xylem as well as the phloem strands, this is called as conjunctive parenchyma. So initially it is of the parenchymatous, but in the older stages, whenever the root is getting older, this parenchyma will become the sclerenchyma just to provide the extra strength and support to this. Now, and one more difference between is dicot and monocot. Whatever this exodermis, cortex and endodermis region, we discussed even in the dicot root, same like the monocot. But in the monocot root, whatever this cortex region is there, that is more extensive compared to the dicot. It is more extensive than the dicot root. And coming to this, same below this endodermis will be there, will be showing all these Casparian strips, Casparian thickenings, which is helpful for the symplastic pathway. Hypoplastic water will be entering the symplastic pathway. So this pericycle will be containing the parenchymatous cells, but uh, this endodermis will contain in the parenchymatous cells, but in the older stages, in the older roots, it will become sclerenchymatous. So they will become sclerenchymatous and they will become more fibrous just to add the extra strength and support to this. And if you are just finding, the center region will be the medulla. Usually in the dicot, we will be having a less or the scanty medulla because of the secondary growth, but here in the monocot, we can find a proper medulla region. So I'll be writing here what type of region it is. So the medulla here is the center part of a stele, and this is surrounded by the vascular bundle. So this medulla is surrounded by vascular bundle. And whenever it is getting older, so you can find a large region of central region called as a medulla. It is made up of parenchymatous cells, and the main function of this medulla is to store the food. In the older roots, whenever the roots are getting older, that time all the parenchyma will be replaced with the sclerenchyma just to provide the mechanical strength and support. So as we are seeing here, these are only the differences between the dicot as well as a monocot. Let us go with this vascular bundle. What type of vascular bundle can be seen here? I'm just writing the description. As the vascular bundles, the separate strands of xylem and phloem are present on different radius and they are jointed by the ground tissue. So that ground tissue initially made up of a parenchyma, later at the maturity replaced with the sclerenchyma. So these vascular bundles are radial and they are separated because they are not on the same radius, not adjacent to each other, and you cannot find any cambium like the dicot root. So as the cambium is absent, these vascular bundles are said to be closer type of vascular bundles. Exarch type of arrangement of primary xylem can be seen. Many strands of xylem are present, more than 6, that's why it is of the polyarch. More than 6 vascular bundles with alternate xylem and the phloem. So these are only the difference between dicot and monocot root. So as there is no cambium, so this is the major important, there is no cambium formation in the case of monocot. So as the absence of cambium, you cannot see the secondary growth. So there is no secondary growth in the case of monocot root. Only the dicot root can show the secondary growth. So all these are, this is the description about monocot and the difference between dicot and monocot root.